I would like to refresh on cestodes, Tapeworms of small ruminants. Mortality is very common in sheep and goats due to various reasons under open grazing system, particularly in young lambs and kids. So many a times mortality is beyond the acceptable level, which is an alarming situation now. So there are many reasons for mortality of sheep and goat. Internal parasites are one of the reasons. So among the internal parasites, helminths are the first cause of illness and deaths beyond the acceptable level in sheep and goats, mainly under 1 year of age, mainly lambs and kids. Parasites in general are grouped as metazoan parasites and protozoan parasites. Metazoan parasites are nothing but multicellular and protozoan parasites are unicellular in nature. So helminths parasites are divided into mainly 3 groups. As you know, first group is nematoda, which includes all roundworms, and second group is cestoda, which includes all tapeworms, and third group is trematoda, which includes all flukes. So here I have given more focus to tapeworms of sheep and goats, as these tapeworms are causing more morbidity and morbidity in sheep and goats. All these tapeworms are segmented endoparasites with an elongated dorsoventrally flattened body, that is tape-like body as shown in the figure, without an alimentary canal. They come under phylum Platyhelminthes. So it is classified into 2 classes, 2 important classes. One is Eucestoda and second one is Cotyloda. So importance of tapeworms, they are only parasites visible in feces and are mainly diagnosed by observing segments, that is proglatids in the feces. Tapeworms can cause very mild unthriftiness and digestive disturbances, intestinal blockage, and can affect gut mortality and also predisposing all the lambs and kids to enterotoxemia disease. So all cestodes are also hermaphrodites, means they are having both male and female reproductive organs, sex organs, as a natural condition or as abnormal body condition. So all these tapeworms depends on its source for their nutrition as well as reproduction. So they are called as obligatory endoparasites normally. So they have elongated flat body without any body cavity. The body is dorsoventrally flat and whitish in color they are, and their size varies from few millimeters to few meters in length. Mainly, a typical cestode usually consists of 3 main body parts in its body. So first part is head or scolex. It is also having also called as acetabulum. It has attachment organs like suckers and hooks. Then second part is unsegmented short region, that is germinal portion. It is called as neck. This neck continuously produces proglatids which constitute the strobila, and these proglatids are separated by transverse constrictions as shown in the figure, and each proglatid consists of 1 or 2 sets of reproductive organs, and third part is strobila, which constitute main bulk of the body and is made up of chain of mature and gravid proglatids. So usually 4 methods of discharge you will observe in cestodes. First one is apolysis, and second one is pseudo-apolysis, third one is para-apolysis, and fourth one is Thizinus condition. Apolysis means eggs in the gravid proglatids are liberated by disintegration of proglatids, known as apolysis. That process is called as apolysis. And pseudo-apolysis means when egg production is ceased, chains of exhausted proglatids may be attached, may be detached from the proglatid. That is called as pseudo-apolysis. And third one is hyperapolysis. Segments shed while they are immature and they lead an independent existence outside of the host. This process is called as hyperapolysis. And Thizinus, this is in this process some tapeworm eggs are released out through an opening created in the uterus, and by the pressure of the muscles, eggs and muscular activity of the proglatid, eggs will be released out. This process is called as Thizinus condition. So these are the 4 important process for release of eggs in the case of cestodes. Then paruterine organs. So in some cestodes, the uterus may degenerate and eggs may either be singly or in groups will pass out into protective structures within the proglatids. So these are called as paruterine organs as shown in the figure 1 and 2. So these these are the paruterine organs showing egg pockets, mainly seen in the case of Diplodium Canine, Stylicia and Avitelina tapeworm infestations. So eggs normally become fully embryonated and contain oncosphere, that is embryo which is armed with 3 pairs of hooks as shown in the figure. So showing 6 hooks, so it is called as hexaconch embryo as shown in this is the first picture showing hexaconch embryo. And during the development of eggs, mainly you will find 4 envelopes around the eggs. Outer thick capsules as shown in the second figure, and outer envelope, inner envelope, and third one is the, and the fourth is the oncospiral membrane. So this embryophore is the principal and more resistant covering of all the TE eggs. So they are more resistant to all environmental conditions. So developmental stage, which comprises of ciliated embryophore containing oncosphere, is called as coracidium. This is the third picture showing the coracidium. So among the helminths, cestodes are the main reason for morbidity in weaned lambs and kids of about 3 to 9 months of age. Hence, control and prevention of all these cestodes is very important to prevent illness as well as mortality between 3 to 9 months of age. So understanding of cestodes, general life cycles, as well as various systems is helpful in proper planning of our treatment and diagnosis. Life cycle of all tapeworms is indirect except Hymnolopis nana, which is called as dwarf tapeworm of humans. On ingestion of eggs by the intermediate host, the oncosphere is released by the action of digestive enzymes in the intestine. So inside the intermediate host, this activated oncosphere emerges out from its oncospheral membrane, and further it will predict deeply and further development will occur. So in the cotyloda, the larva is called as coracidium, and it has to be eaten by intermediate host. So cysticercae develop in different parts like striated muscles, brain, eyes, spinal cord, skin, etc. So those cysticercae which are present in the muscles usually ingested along with the raw or undercooked meat or pork. Further development will occur in the definitive host. So adult tapeworms live in the small intestine and they liberate the eggs and proglatids through the feces of the host, intermediate host. So the cycle continue. Human beings will be infected occasionally if they eat the raw or undercooked meat. So cysticercae begin degenerate in about 4 to 6 months. By 9th month, the most of the infected intermediate hosts like sheep and goat will die if you are not treating properly. I will discuss about various larval stages of cestodes. So in cestodes, different larval forms develop inside the intermediate host and are collectively called as bladderworms or metacestodes. So these larval stages are called as bladderworms or metacestodes. These develop both in invertebrate as well as vertebrate host. First larval stage of tapeworm is called as cysticercus, a larval form with a single invaginated scolex into a large fluid-filled bladder. As shown in the figure, this is the single scolex invaginated into a large fluid-filled bladder. This is called as cysticercus, mainly seen in the case of Ctenidae family, mostly observed in all striated muscles of intermediate host. So you can observe in the first picture, these are the cysticercus larvae present on sheep pig muzzle. The fourth picture, you can observe the removed tinea cysts. Second metacestode is called a cysticercoid. Cysticercoid means a pinpoint sized vesicle with an evaginated scolex. In cysticercus, it is invaginated, whereas here it is evaginated scolex. So into a small solid cyst is called a cysticercoid larvae. It develops mainly in invertebrate hosts, mainly in the case of ruminant, equine, and poultry tapeworm infestation. In the case of Anoplocephalida class. So third larval stage is called a Sinuous. It is Sinuous, a thin semi-transparent bladder containing plenty of fluid with a large number of scolices invaginated from its inner genital layer, as shown in the figure. So this is the inner genital layer from which a large number of protoscolices invaginated. So in the picture, you can observe the infected brain of sheep showing Taenia multiceps metacestode. So under microscope, it will appear like this. This is the separated metacestode from the brain of sheep. So fourth metacestode stage is called a strobilocercus, an evaginated scolex attached to a bladder by a segmented strobila. So this is the scolex and this is the bladder. So these two are attached by segmented strobilas. Hence this name strobilocercus. So this is mainly seen or observed in the case of Taenia teniformis infections. So in the second picture, you can observe the presence of strobilocercus larvae. This is white color larvae present in the livers of rodents. Rats infected with Taenia teniformis infection. So fifth metacestode is hydrated, a thick-walled, large-sized bladder filled with fluid, producing a large number of brood capsule from its inner genital layer. Inside each brood capsule, again you can find a large number of protoscolices. So this is called hydratissis. These hydratissis may be identified in different organs of ungulates, sheep and goat and man, and during mainly due to Echinococcus granulosus infection. So all these detached scolices and brood capsules found freely sometimes in the cystic fluid, that condition is called as hydrated sand. So this is the brood capsule having many protoscolices inside. means originated from its inner genital layer. This is all the fluid. So these are the separated cystic circus larvae. Next slide is the next metacestode is Tetrathyridium. It is a solid worm-like elongated larvae with deeply invaginated acetabular scolex. So scolex is invaginated inside the larvae. So this is the second stage metacestode commonly observed in infections in the case of reptiles and mammals. Then next metacestode is Cryptocystis. So a cystic in which tail is present is called as Cryptocystis, and it is found mainly in case of early development in arthropod intermediate hosts, mainly seen in arthropod intermediate hosts. So all these are the definitions. And Coracidium, egg with 6 hooked oncosphere covered with a ciliated embryophore is called as Coracidium. This is the Coracidium. And a solid-bodied metacestode with hooks in the segmented section or constricted area is called as procercoid. And this is pleurocercoid. It is the next stage to procercoid. It is an elongated solid-bodied metacestode which bear an adult scolex. So well-developed scolex is there in the case of pleurocercoid. So Coracidium, procercoid, and pleurocercoid, all these larval stages will be found in the case of Diphyllobothium latem infections. So this slide shows comparison between different bladder worms of sheep and goat. So first one is Taenia hydratigena. It will second one is Taenia ovis, third one is Taenia multiceps, and fourth one is Echinococcus granulosus. So Cystocircus tenicolis is the bladder worm of Taenia hydratigena, mainly located in the liver, omentum, and mesentery. It measures about 25 to 30 mm in size. And second larval stage is Cystocircus ovis. It appears like a small oval sac-like structure filled with clear fluid, and it mainly measures up to 10 mm in size. And it is mainly observed on heart, diaphragm, tongue, and mastication muscles, and walls of esophagus, and walls of stomach, and other muscles. Then Sinuous, this is a bladder filled with many scolices. This metacestode is the larval form of Taenia multiceps. It is also called as multiceps multiceps. It measures to a size of 10 egg size or sometimes more in size, and mainly located in brain and spinal cord. This Sinuous is mainly located in brain and spinal cord of sheep and goat. Then hydrated cyst. This is the metacestode of Echinococcus granulosus, which mainly measures about 2 to 6 centimeters in size and mainly located in liver and lungs. So normally we found two types of hydrated cysts in the animals. One is fertile cyst and the other one is sterile cyst. Fertile cyst can able to produce brood capsule with protosclerosis, whereas sterile cyst means they are unable to produce protosclerosis. So this slide shows details of treatment measures as well as PM findings and control measures of important bladder worms of sheep and goat. No effective treatment is available up to now for the control of these bladder worms. So hydratissis will be having zoonotic importance. So it is better to educate humans about these bladder worms for the control of this hydrated disease. Next chapter is on ruminant tapeworms. So major ruminant tapeworms of sheep and goat are Menesia expensa, Menesia benedini, Tevetelinus, Stelicia, Thysinesia, and Thysinosoma actinides. So all these ruminant tapeworms are large and their segments are passed out in the dung. So adult tapeworms mainly found in small intestine of sheep and goats, except Stelicia hepatica, which is mainly located in bile ducts. So of all these tapeworms, most common tapeworm of sheep and goat is Menesia, and it is also called as broad tapeworm. The adult worms cause few symptoms in definitive host, but their larval stages or metacestodes will be causing considerable damage to sheep and goat intermediate hosts. So Menesia expensa, commonly called as broad tapeworm, and Thysinosoma actinides, also called as fringe tapeworm, and Echinococcus granulosus causes the hydrated cyst, which has a zoonotic importance, and Taenia ovis and Taenia hydratigena causes cystocirrhosis, and Taenia multiceps is causing git or circling disease. So Avatelina centripunctata is the first tapeworm, mainly located in the small intestine of sheep. They are long, narrow cestodes with distinct segmentation. They occur mainly in small intestine of sheep and goat and other ruminants. There is a single set of genital organs with irregularly alternating genital pores in short proglottids, as shown in the first figure, and vitelline and shell glands are completely absent, hence the name Avatelina to the genus, and uterus is mainly transverse and are placed in the middle portion of each proglotid as shown in the figure, and eggs then passed into thick-walled paruterine organs in which egg lay in capsules as shown in the figure 2, and uterus and paruterine organs will be shown as opaque line in the middle portion of each proglotid, which is the characteristic identification feature of this Evetellina tapeworm under microscope. So there will be a opaque line in the middle of each proglotid. This is the identification feature of Evetellina proglotid, and the eggs will have no pyriform apparatus, and life cycle is indirect, involving bark lice, dust lice, and book lice as intermediate hosts. And second tapeworm, sheep tapeworm, is Stylicia species. These are long and narrow cestodes with a distinct segmentation as shown in the figure, and they are located mainly in the small intestine of ruminants. There is a single set of genital organs. Testes are few, situated on the lateral aspects are fields, and ovary lies towards the oral side. And uterus first appears as a long transverse dumbbell-shaped tube and later replaced by two paruterine organs as shown in the figure. And life cycle is indirect, including oribatimates as intermediate host. So this is very important. Oribatimates acts as intermediate host for Stylicia globipunctata tapeworm. So this slide shows the pathogenesis caused by Stylicia globipunctata, and scolex of immature worms mainly penetrates the intestinal mucous membrane of sheep and goats and forms nodules and causing the disease nodular teniasis, or sometimes sheep measles it is called, with proliferative inflammation and cellular infiltration and epithelial desquamation as shown in the figures. So this is the pathogenesis caused by Stylicia globipunctata in case of sheep. And the Thysanosoma actinides is the third tapeworm, and it is commonly called as fringed tapeworm of ruminant. It occurs in the bile ducts, pancreatic ducts, and small intestine of sheep and causing damage. So segments are short and conspicuous, fringed posteriorly as shown in the second figure. So they are fringed. And it is another double port tapeworm of ruminants besides Monesia. And as each segment contains two sets of reproductive organs, it is called as double port tapeworm. And life cycle is indirect, with the lice acting as intermediate host. So for Stylicia, it is oribatimae, whereas for Thysanosoma, it is lice. So cysticercoids normally develops in the posed lice, and these are ingested along with the vegetation for further development in the host. And fourth one is Thysanesia giardi, mainly located in the small intestine of sheep, goat, and cattle. It is also called as Cytomectra. And segments of this tapeworm are short and contain single set of genitalia, and testis lie lateral to the excretory canal, and side of this segment will be having serous sacs. So this bulges out and thus giving the irregular appearance to the tapeworm, the margin of the tapeworm. So genital ports are irregularly situated alternately, and eggs will pass from the uterus into a large number of paruterine organs, and pyriform apparatus is absent in the eggs of this Thysanesia giardi tapeworm. And next important tapeworm, that is Monesia, mainly located in the small intestine of sheep and goat. It is also called as double port tapeworm of sheep or ruminant. So it has four suckers in its morphology. In the scolex without any hooks, so it has unarmed scolex. So scolex without any hooks is called as unarmed scolex, and segments are wider with bilateral genitalia, hence the name double port tapeworm of the sheep. Posterior border of each proglotid will have a row of rosette-like interproglotidal glands, and eggs are triangular in shape with well-developed embryo inside, that is pyriform apparatus. The eggs are triangular in shape, as I will show you in the next picture. It is showing infections in young animals, and their incidence is coinciding with the active periods of vectors, mainly oribatimates. You should remember it. So the Monesia species are commonest in young animals, and their incidence is coinciding with the active periods of vectors, mainly oribatimates. So normally you find two species under genus Monesia. First one is Expensa and second one is Benedini. So Monesia expensa is commonly found in the small intestine of sheep, whereas Benedini is found in the case of cattle. So morphology of Benedini is similar to that of Expensa, but differ slightly in the distribution of interproglotidal glands. So interproglotidal glands are confined to a short and continuous row in the middle portion of segment in the case of Benedini, whereas as shown in the figure, and whereas in the case of Expensa, all these glands are extended all along the full breadth of the proglotids. So egg is square shape or quadrangular in shape in the case of Expensa, whereas egg is triangular in shape in the case of Benedini, as shown in the pictures on the right side. So this is Expensa egg and this is Benedini egg. These are the differentiating point. Now we will see the life cycle of Monesia expensa. This is indirect, involving overweighted mites as intermediate host. So mature proglatids are mainly voided in the dung of definitive host. Eggs are eaten by the overweighted mites, which acts as intermediate host. So cystisarcoids normally develop in the grass mites. Definitive host gets infection by the ingestion of these infected mites. And proglatids and the eggs will be passed out in the feces of the infected animals, and these proglatids may be eaten by sometimes birds, and which therefore may disseminate the infection. And so adult tapeworm will develop in the small intestine of ruminants, and the cycle will be continuing. So this is the life cycle of Monesia. Birds acts as accidental hosts. They will be disseminating the infection, and overweighted mites mainly acts as intermediate hosts. What is prepatent period? It is a period between the infection with a parasite and production of eggs by a female worm. Is called as mainly prepatent period, and generally cestodes may take 35 to 40 days as a prepatent period for their development, and this period is equivalent to the incubation period of microbial infections, but biologically differs because the parasite is going through various developmental stages in the definitive host. Auto-infection. What is auto-infection means? Usually occurs in individuals with impaired cell-mediated immunity. So the time between egg being deposited on the pasture and development of infective stage in a host, that is sheep, is called as auto-infection period. Mainly it is about ranging between 60 to 150 days in the case of cestodes. Then pathogenesis of Monesia. The disease is called as monesiasis. Young animals like lambs, kids are substantially infected. In heavy infections, intestines may have solid mass of tapeworms, which cause obstruction. It may partly obstruct the flow of bile ducts, bile, and as well as pancreatic juices, and cause digestive disorders, unthriftiness because of presence of this overload of these tapeworm. There may be stasis of intestinal movements, which causes anaerobic situation in the intestines. So this anaerobic situation may favor multiplication of Clostridium bacteria, which is natural inhabitant of intestines, leading to enterotoxemia in the case of young lambs. So young animals will show some clinical signs like diarrhea, unthriftiness, emaciation, anemia, decreased wool and meat production, and you will find normally gravid proglatids in the feces of all these infected lambs. So in the pictures, you can observe the diarrhea, and this is the adult worm, Monesia expensa. So because of overload of all these tapeworms, there may be stasis of intestinal movements, so which causes anaerobic situation. So this may favor the multiplication of all Clostridial anaerobic bacteria, which is the natural inhabitant of all intestine, leading to enterotoxemia. So this is the predisposing condition, monesiasis, in the case of lambs for enterotoxemia. So now how to diagnose this monesiasis? Mainly based on history and clinical symptoms shown by the animals and presence of ripened segments by the adult worms. Mainly they resemble cooked rice grain like appearance, and confirmative diagnosis is mainly through finding of characteristic eggs, that is triangular shaped eggs in the case of Monesia benedine infection, and quadrangular shaped eggs in the case of Expensa infection. And these eggs will be having well-developed pyriform apparatus as shown in the figure. This is the egg with the pyriform apparatus inside the egg. Fourth feature is finding of adult worms in the intestines of died sheep during post-mortem. So this is one of the options for the correct diagnosis of monesiasis. So recovering of adult worms from the intestines of died sheep is the most correct diagnosis for identification of Monesia infection in case of sheep. So all these are the treatment drugs for treating of this monesiasis, but combinations are recommended for mixed infection of cestodes and nematodes. Normally, all benzimidazoles like albendazole, fenbendazole, and mebendazole are showing broad spectrum of action, and their action is mainly related to their therapeutic efficacy and concentration in the blood of infected animals, and they kill the cestodes through disruption of energy mechanism by binding to a protein required for uptake of nutrients. Since some benzimidazoles like albendazole, mebendazole are teratogenic, cancerous, so they should not be used in early pregnancy. And fenbendazole can also be used even in pregnancy, but we found lot of antihelminthic resistance in the field condition. So niclosamide and praziquantel and the albendazole are the safe drugs to be used by all the sheep farmers for the control of these tapeworm infections. So we can also use another drug, that is ericoline hydrobromide. It is called as Pachivakka in Telugu. It can be used at a dose rate of 5 mg per kg body weight as safe and economical drug while grazing of these sheep in forest lands. And niclosamide and butanol drugs are also can be used, but these are more costliest drugs. Now, in shops, niclosamide plus albendazole combination tablets are available. They are efficiently working on dog tapeworms. These are some of the old remedies, oldest herbal drugs which are used for the treatment of tapeworm infection in sheep and goats during olden days. So they are showing anti-cestodal activity against adult worms, but not showing action on metacestode stages. So now we are using other drugs like praziquantel and albendazole. So how to control and prevent this monoziasis? Mainly control and prevention of this monoziasis is through keep away of all stray dogs from eating of carrions or other raw meats in the farms, and treatment of all infected animals by periodical deworming with albendazole and praziquantel combinations, and control of intermediate host by regular ploughing and reseeding of infected pastures to prevent the mites infestation and lice infestation, and proper disposal of sheep feces, and avoid pasturing by our small ruminants in the early mornings. Why? Because when there is peak, there will be a peak activity of all these forage mites in the on the grass. So regular testing of pastures for the presence of forage mites, the control measure to be followed to prevent this, for the control of this monoziasis disease in sheep. We will discuss about dog tapeworms. So Diplodium caninum, Taenia hydratisina, Taenia ovis, and cerealis, and Pisciformis multiceps, and Echinococcus granulosus, and Diphyllobothrium latum are the common dog tapeworms. These dogs acting as definitive host, but our small ruminants like sheep and goats acting as intermediate host. So infections with adult stages are not very harmful to the dogs, but they are more harmful to our ruminants. So of all these Diplodium caninum, multiceps, ovis, and Echinococcus granulosus and hydratissis are causing serious condition in our ruminants. So I will discuss in detail about some of these important tapeworms. So Diplodium caninum, it is most common and cosmopolitan parasite of dog, cat, fox, and occasionally children. Coming to its morphology, it has two sets of genital organ in each probiotic, hence the name double-pore tapeworm of dog. The ovary and vitelline glands will form a mass on either side of the proglotid resembling bunch of grapes. So this is another identification character of proglotids of Diplodium, and mature and gravid proglotids will have a characteristic elongate shape resembling cucumber seeds as shown in the figure on the top right side. So this is the figure showing the cucumber seed shaped proglotids of Diplodium. Life cycle is indirect, involving fleas and lice as intermediate host. So dog will process gravid proglotids through feces, and they may leave the host spontaneously, and they actively crawl actively and disseminating the eggs throughout the field. So the intermediate hosts are dog fleas and louse. So the larval flea or louse ingest the eggs, and cystic sarcoid will normally develop in the adult insects, and definitive hosts are infected by accidental ingestion of these oribitid mites or lice. So intermediate hosts are normally dog fleas and louse. So the larval flea or louse will ingest all these eggs, and cystic sarcoids develop into adult insects. So the definitive host normally infected by swallowing of these infected flea and louse. Humans accidentally will get the infection by accidental ingestion of these infected fleas while fondling with the pets. Disease pathogenesis caused by this is called as diplodiosis. Pets with heavy infection will show diarrhea, constipation, weight loss, restlessness, abdominal pain, and pot-bellied condition due to itching caused by these migrated gravid probiotids present in the anal region. The dog draws its anus onto the ground. So anal pruritus is the most common pathognomic symptom in the case of dogs infected with this diplodiosis. And diagnosis is mainly by finding of cooked rice grain like gravid probiotids in the feces, and confirmative diagnosis is by finding of characteristic eggs that in egg pockets when seen under microscope. So this is the picture showing the egg packets under the microscope. So dog showing the anal pruritus in the first picture. So these are all the identification features of Diplodium caninum. First and last picture will be showing the cucumber seed-shaped gravid probiotids of Diplodium caninum. When dry, they appear like cooked rice grain. So this is like cooked rice grain. So they are often all these segments are often passed out in the feces of the infected dog. In the second picture, you can observe the egg pockets of Diplodium caninum in the fecal examination. So this is the egg packets showing many eggs. And in the third picture, this picture, you can observe the presence of adult Diplodium caninum under the mucosa of intestine during post-mortem examination. So praziquantel is very effective drug for both adult and immature stages of tapeworms, whereas albendazole is also more effective under benzimidazole group of drugs. So benzimidazole antihelminthics only kill the head and segments, and it will be less active when compared to praziquantel. So it is better to use both combination of albendazole and praziquantel for the control of all the tapeworm infection in small ruminants. So control of this diploidiosis is mainly through flea control. Next tapeworm is Taenia hydratisina. It occurs in the small intestine of dog and wild carnivores, and sheep acts as intermediate host for this tapeworm, and eggs are oval in shape, and this larva is called as Cysticercus tenuicolis. It causes a disease called as hepatic cysticercosis in the case of sheep. So prevalence of this Taenia hydratisina in sheep is very high, and occasionally young lambs will suffer more, and it causes traumatic hepatitis leading to death of the sheep, which may be confused with that of acute fasciolasis. But oozing of blood from the nostrils will not be there in this infection, this tapeworm infection. Whereas in the case of acute fasciolasis, you will observe oozing of blood from the unclotted blood from the nostrils. Infected lambs will show unthriftiness, abdominal problems, migrating cysticercus in the larvae in the liver may cause hemorrhagic and fibrotic tracts in the liver. So viable caseated cysticercus may be present on the liver as shown in the figure. So this condition is called as hepatic cysticercosis, and it will result in condemnation of liver during slaughter. Next tapeworm is important tapeworm, that is Taenia multiceps. It is also called as multiceps. The adult worm grows in the small intestine of dog, fox, coyote, and jackal. The metacestose stage is called as Sinuous cerebralis. It mainly develops in the brain and spinal cord of sheep and goat and ungulates. It is also recorded in man. So this bladder worm has numerous colicus attached to its inner wall, as shown in the figure. And infective stage is mainly seen in the this bladder worm is mainly infective to intermediate host, mainly sheep and goat. So life cycle of this Taenia multiceps is indirect, involving ruminants as intermediate hosts. So cysts will be found in different organs. So we see the pathogenesis in lambs. A large number of immature stages are sinuae. They may migrate to brain, causing destruction of brain tissues of sheep, resulting in meningoencephalitis. Based on location of the cyst, clinical symptoms of multiceps may vary, and the condition is known as git, staggers, or studdy, or high-stepping gait. If the cyst is located in the parietal surface of one of the cerebral hemisphere, then the sheep holds its head to one side and its opposite eye becomes blind. If the cyst is located in the anterior part of the brain, sheep presses its head against the chest, and animal steps high and may walk in straight line until it gets stopped by an obstacle and remains in a stiff gait for some time or some period. If the cyst is located in cerebellum, then sheep show hyperesthetic movements and jerky and staggering gait. If the cyst is located in the spinal cord, then the sheep shows progressive paresis of one or both the hind limbs. If the cyst is located on the surface of the brain, then skull undergo pressure atrophy, and there will be perforation of the skull will be seen. So these are the photos of sheep infected with git. So in the first picture, you can observe the lateral deviation of the head and showing circular movement. And second picture, you can observe the pressing of sheep head against the wall or hard object. In the third picture, you can observe the incoordination and ataxia. Ataxia means loss of full control of body movements and falling down. And fourth one is the lateral recumbency with stretched out hind limbs of the infected sheep. So all these are the symptoms of GID. So this is the one research paper on non-cerebral sinuosis in case of sheep, presented by me recently to our SVU booklet. Usually these sinuous found in various parts of brain and spinal cord. Sometimes sinuous cysts may be present on muscles of thigh, shoulders, neck, diaphragm, tongue, and intercostal and skeletal muscles. So those cysticercae which are found other than the CNS parts, this condition is called as non-cerebral sinuosis. So metacestodes of canine tapeworms are causing considerable mortality and causing financial losses to the small ruminant farmers. As a result, carcass contamination will be there during slaughter. So a sheep, so this is the case of non-cerebral sinuosis. A sheep with swelling on both the sides of mandibles was presented with a history of pain, unable to chew grass from 7 days onwards, looking dull and weak. On palpation, we found out that it is a cyst. The removed cyst we removed cyst surgically, and cyst was identified as non-cerebral sinuosis. Cyst based on morphological characteristics like presence of bladder filled with watery fluid, having thin, fibrous, and semi-opaque transparent surrounding membrane with numerous calyces attached to its inner surface. So these are the two cysts removed from both the sides of the mandible. So diagnosis is mainly done by cystic fluid analysis. So finding of monocytes and neutrophils in the case of cystic fluid using spectrophotometer, and associated clinical signs indicating the presence of sinuous cerebral cyst in non-CNS parts of sheep. So this condition is commonly caused by Taenia multiceps in the case of sheep, whereas Taenia gaigeri in the case of goats. So these metacestodes are causing a disease condition, mainly infecting intramuscular and subcutaneous tissues of sheep and goats. No special treatment is there for sinuous condition except correct identification and removal of cyst by surgical intervention, and periodical deworming is necessary. Deworming of all sheep along with stray dogs in the case of sheep flocks, and proper disposal of infected carcasses are the control measures to be followed for the control of these sinuous. Next important tapeworm is Echinococcus granulosus. So species of Echinococcus are very small cestodes, usually possessing 3 to 4 segments or proglatids as shown in the figure. If you come to the morphology, this form possesses only 3 to 4 proglatids. The penultimate proglatid is mature proglatid, and terminal proglatid is called as gravid proglatid, and is usually about the half the length of the tapeworm, whole tapeworm, and the rostrum has 2 rows of hooks. The ovary is kidney shaped, and the ovary is kidney shaped, and genital pore is located near the middle of mature and gravid proglatid, and the gravid proglatid usually disintegrate in the intestine, and therefore only eggs will be found in the feces. They become immediately active, and adult worms may mainly located in the small intestine of man, dog, and ungulates. So larval stage is called as hydatid, causing hydatidiosis condition in the case of small ruminants, sheep and goat. So these are this slide is showing different species present under genera Echinococcus. Mainly 4 species were found. Echinococcus granulosus is the first species causing cystic echinococcus in ungulates and men, with dogs and other canids acting as definitive host. Whereas Echinococcus multilocularis, this is the second species, and causing alveolar echinohydatidiosis, with foxes acting as definitive host and rodents as intermediate host. Echinococcus oligotheris is causing polycystic hydatidiosis, with wild ferrets acting as definitive host and rodents acting as definitive in the intermediate host. Echinococcus vogeli, it is also causing polycystic form in the case of domestic dog and bush dog. Then we will see the life cycle. It is indirect, involving intermediate host, that is sheep and goats, ungulates and man. Man is the accidental host. They may get infection through eating of contaminated feed or water, forage contaminated with the feces of infected dogs. So eggs will be passed out in the feces of carnivores. After ingestion by the ungulates, the oncospheres will penetrate the intestinal mucosa and venule, and as well as lymphatics. They reaches the liver and lungs, and the hydratissis develops slowly over several months, and humans develop infection through intimate association with the infected dogs, particularly children, while fondling with the pets. So both pastoral as well as sylvatic. Pastoral means domestic animal involvement, and sylvatic life cycle means wild animal involvement will be there in the life cycle. So these are the hydratissis, commonly 5 to 10 centimeter size, principally recorded in man and sheep. So lungs and liver are the most common sites where you will find hydratissis. In sheep, hydratissis commonly found in lungs, and they are multilocular in nature. Not all hydratissis, they will produce brood capsules. So those cysts are called as sterile cysts. Sometimes brood capsules become detached and float free in the cystic fluid. Such condition is called as hydatid sand. And in sheep, 49% of cysts are fertile, whereas 51% are sterile. So maximum fertility is observed in sheep when compared to other animals. So sheep are considered as suitable intermediate host for Echinococcus granulosus. So this point should be remembered. So sheep are considered as suitable intermediate host for Echinococcus granulosus. In case of cattle, normally, frequently you will find this sterile cyst, whereas in sheep, 51% of cysts are sterile and 49% are fertile. So sheep are considered as more suitable host for Echinococcus granulosus. If you take the cystic fluid, it is very pale in color with 17 to 200 micrograms of protein, will be there for every 100 ml of cystic fluid, and this fluid will have striking similarity with that of the serum of the host, as it contains many immunoglobulins, some anti-complementary substances, as well as calcareous substances. All these elements are associated with the long survival of these hydratissis in the host. So pathogenesis mainly depends on severity of infection and location of cyst on various internal organs like brain, heart, lungs. If the cyst is located in brain and heart, the function of these organs will be frequently impaired. In domestic animals, clinical signs are usually seen in heavy infections only. So rupture of cyst will cause leakage of this hydrated fluid, which can lead to various allergic manifestations like urticaria, asthma, anaphylactic shock, as well as death. So diagnosis is mainly through fecal examination for the presence of Echinococcus eggs, but eggs of Echinococcus cannot be differentiated from other teenage eggs such as Taenia solium and Hydratissis. So confirmation is mainly based on demonstration of adult worms during post-mortem examination. So for the collection of adult worms, all the dogs must be treated with auriculine hydrobromide at a dose rate of 1 to 2 mg per kg body weight. All the treated dogs will purge out the intestinal contents and expel all the adult worms. So examine the mucous portion of the fecal sample to obtain the adult worms. So radiographic examination is also helpful for detection of this hydratissis in sheep. In bovines, hybridoma technique is also used, and radiographic examination is also helpful, and ultrasound examinations, ultrasonography, and imaging are the immunodiagnostic tests which are useful for detection of this hydratissis in human beings. Prevention and control. Treatment is mainly surgical removal of hydratissis and deworming with mebendazole in case of sheep and humans, and control is mainly through. Many countries are endemic, so control and prevention strategies of cystic echinococcus is aimed at intervening at certain points of parasite life cycle in case of dogs. So many countries are endemic, so control and prevention strategies of cystic echinococcus mainly aimed at intervening at certain points of parasites life cycle in case of dogs, in order to discontinue the life cycle. And deworming of dogs and cats periodically, and proper immunization of dogs with irradiated protosclerosis of Echinococcus granulosus at 30,000 to 60,000 rads of intensity was reduced the development of time of cystode, and sex organs of all the cystodes remained immature in the treated animals, according to Amin Janov et al. 1980 research. The secretory and excretory antigens derived from the adult Echinococcus granulosus in vitro has shown limited success. So proper disposal of all infected carcasses and offals is also recommended, and some research showed that treatments with niclosamide showed no benefit in terms of weight gain, fecal consistency between the treated and untreated lambs. This is all the research part of Echinococcus. And this is another paper submitted by me on tapeworm infection in organized sheep farm and control measures, published in International Journal of Scientific Research, Pakistan. So prevention and control of program of sheep and goat tapeworms in endemic areas will improve the production potentials of tapeworm infested in small ruminants for economic well-being of these marginal farmers. So there is a need for further investigation using molecular techniques for accurate identification of carrier status of Sinuous infection in the case of small ruminants. So to summarize this webinar, mature tapeworms like Monogia species are mainly located in the small intestine of sheep and goats and causing serious disease in sheep, and bladder worms like Cystococca stenicolis causing sheep measles, and Sinuous cerebralis causing GID, and Echinococcus granulosus causing hydratissis are some of the important bladder worm infections of small ruminants. So, for overall treatment of adult tapeworms, niclosamide can be used at a dose rate of 50 mg per kg body weight, and praziquantel can be used at a dose rate of 10 mg per kg body weight. For bladder worms, no effective treatment is available except for Sinuous cerebralis. We can try usage of praziquantel up to 50 to 100 mg per kg body weight. So, prevention and control of tapeworm infections is mainly through breakage of reinfection. It can be done through prevention of eating of infected mites present on the grass blades and soil by the small ruminants, which is the more crucial to prevent the tapeworm infection in small ruminants. And this can be done through zerograzing of all weaned lambs and kids. And second thing is to avoid keeping of dogs, particularly stray dogs, along with the sheep flocks. as they will be disseminating infection through defecating on grazing lands. And third point is through periodical deworming of all sheep and goat present in a flock at a time. And last point is preventing eating of raw meat or offals by dogs. It is to conclude that tapeworms will pose a major threat to our small ruminants. So it is imperative to safeguard our sheep and goat from tapeworms like Monesia, as they are causing serious health problem. So for the control of ruminant tapeworm, it is better to go for benches containing the albendazole and praziquantel, which causes successful removal of all these tapeworm segments under field conditions. So Echinococcus granulosus is another important tapeworm causing hydratic disease in livestock and humans. So because of all this of its zoonotic importance, it is better to educate all dog owners and children to adopt effective management strategies like proper washing of hands after fondling with their pets, and deworming of all stray dogs present in the flocks along with sheep and goats, and keep away of dogs from eating of carrions or offals. Or these are some of the important management practices to prevent easy spread of tapeworm infection in farm. So cysts of Taenia multiceps and ovis will be causing contamination of carcasses during slaughter. Resulting in economic losses to the small ruminant farming community. So following deworming regularly for small ruminants after obtaining epidemiological data, this is very, very important to prevent tapeworm infection in endemic areas. Thank you.